Hello everyone, thank you for joining me today. My name is Dr. Madhuri Bandaru. I'm a postdoctoral researcher from Dr. Reddy's Lab, and today I will be discussing the role of RLIP, a protein that has recently gained attention for its potential impact on aging and neurodegenerative diseases, particularly Alzheimer's disease. Alzheimer's is a condition that causes memory loss and confusion and affecting millions of people worldwide. Most research so far has focused on things like amyloid plaques and tau proteins, but we are starting to realize that other factors might also play a role. One of these factors is RLIP, a protein that helps with important functions in the brain cells. In the presentation, I will discuss new research showing a connection between RLIP and AD. Especially, we will look at how changes in RLIP levels might impact brain function and contribute to the development of the disease pathology. By the end of this presentation, I hope you will understand why RLIP could be important for understanding AD and how it might help in developing new treatments. As we all know, AD is a neurological condition that affects the brain, gradually impairing memory and thinking. It is the most common cause of dementia in older adults, and it is recognized by several key symptoms such as difficulty remembering recent events or asking the same questions over and over, getting lost easily or misplacing things, trouble finding the right words or organizing thoughts and plans, issues with balance or putting on clothes properly, increased feelings of worry, anger, or aggression, changes in mood and vision or coordination, confusing day and night, inability to recognize objects or use them correctly, gradual loss of everyday skills, and the ability to perform routine tasks. These are the most common symptoms of Alzheimer's disease patients. AD is a progressive and irreversible disease, meaning that the damage it does to the brain is permanent. There is no known cure, but medications and management strategies can help temporarily relieve from symptoms. Now, coming to risk factors of AD, the biggest known risk factor for AD is age, and most people with the disease develop it when they are 65 or older. However, the exact causes are still unclear and likely involve a mix of genetic, health, and lifestyle factors. Other common risk factors are include family history, having a parent or sibling with Alzheimer's disease increase the risk of developing it. Some medical conditions like cardiovascular diseases, diabetes, high blood pressure, high cholesterol, hearing loss, and depression also increase the risk of AD. Some lifestyle factors that may increase the risk of AD like physical inactivity, unhealthy diet, alcohol misuse, smoking, not getting enough sleep, social isolation, and lack of mental stimulation. There are mostly two types of Alzheimer's disease that includes early onset and late onset. In early onset, this type of disease develops before age 60 and can start as early as person's 30s. It affects less than 10% of people with AD. Some cases are caused by an inherited gene change, while others may be caused by other genetic factors. When coming to late onset, this is the most common type of Alzheimer's affecting people who are 60 and older and typically manifest in their mid 60s. People with one version of the apolipoprotein E gene on the 19th chromosome have an increased risk of developing this type of AD. AD generally advances in three stages: early, middle, and late. However, symptoms can vary from person to person, and individuals may move through these stages at different speeds. In the early stages of Alzheimer's, people may have memory problems, make poor decisions, and show small changes in their personality. As the disease progresses, memory and language issues get worse, and patients struggle with everyday tasks like managing finances or remembering to take their medications. They may also have trouble with visual and spatial tasks, such as finding their way in an unfamiliar place. In the later stages of AD, patients start to lose control over their motor functions. Eventually, they may no longer recognize family members or be able to speak. As the disease advances, it also impacts the person's emotions and behavior. Now, coming to pathological changes, AD is marked by two key abnormalities in the brain: amyloid plaques and neurofibrillary tangles. These are considered the hallmark signs of the disease. Amyloid plaques are found between nerve cells in the brain tissue, and these plaques are formed by when a protein called amyloid precursor protein is broken down by enzymes called beta secretase and gamma secretase. This cleavage produces a fragment known as amyloid beta, which accumulates to form plaques. Neurofibrillary tangles are found inside of neurons, and these tangles are made up of tau protein. In healthy neurons, tau helps stabilize microtubules, which are important for cell structure and transport of materials within the neuron. In AD, tau becomes abnormal, changing shape and twisting into helical filaments that form tangles. These tangles cause the microtubules to disintegrate, disrupting the neuron transport system. As a result, communication between nerve cells becomes impaired and the neurons will die. Other pathological features include alterations in mitochondria, oxidative stress, glial and inflammatory responses, synaptic failure, and neuronal loss. RLIP, also called RALBP1, is a multifunctional protein found in the cytoplasm of cells. It interacts with small molecules called RALA and RALB, which are important for regulating cellular functions like transporting substances, maintaining cellular structure, and controlling cell survival and death. RLIP also acts as a multi-drug resistant protein, helping move various molecules, including drugs, lipids, and stress-related molecules, into and out of cells. One of its key roles is transporting glutathione and other antioxidants which protect cells from oxidative stress, a major factor in aging and diseases like Alzheimer's. Additionally, RLIP helps detoxify harmful compounds by converting them into safer forms that can be eliminated from the body. Majorly, RLIP is a 655 residue protein with two main functional regions, ROGAP domain and RAL binding domain. The ROGAP domain is present between the residues 184 to 368, involved in the activation of small GTPases like CDC42 and RAC1. The RAL binding domain, which binds to RALA and RALB, is present between the residues 393 to 446. The protein also contains two ATP binding sites, enabling it to transport molecules. The N-terminal region is involved in receptor-mediated endocytosis, while the C-terminal region interacts with PSD95 in postsynaptic neurons, contributing to synaptic endocytosis. With this complex structure and several functional domains, RLIP involved in membrane dynamics, cytoskeletal motility, and the RAL signaling pathway, which affects cell growth and movement and may even be linked to cancer metastasis. RLIP found throughout the body, including the brain, where it plays an important role in regulating different cellular functions. Now coming to lipid peroxidation and its impact on Alzheimer's disease, lipid peroxidation is a harmful process triggered by factors such as environmental pollutants, smoking, chronic inflammation, and other AD risk factors. Given the brain's high lipid content, lipid peroxidation is especially damaging to the brain tissue and becomes a prominent feature in late-stage AD and other neurodegenerative diseases. Under normal condition, RLIP removes electrophiles through MAP pathway. The mercapturic acid pathway plays an important role in detoxifying harmful compounds. It involves a series of enzymatic reactions where glutathione S transferase first conjugates a foreign compound with glutathione to form a glutathione S conjugate. This is further processed by enzymes like gamma glutamyl transferase and dipeptidases, which removes additional residues, eventually producing mercapturic acids that are excreted in urine. This pathway helps remove both xenobiotic and endobiotic harmful compounds from the body. RLIP is crucial in the transport of glutathione S conjugates and other conjugates, helping protect cells from oxidative stress. RLIP interacts with the clathrin-dependent endocytosis complex, facilitating the internalization of hormone receptor complexes. It also helps expel harmful glutathione conjugates such as glutathione hydroxy-2-nonitol, which reduces oxidative stress and prevents the suppression of glutathione S transferase, an important enzyme in the glutathione system. Now coming to the connection between RLIP and AD. Generally, amyloid precursor protein is synthesized in the endoplasmic reticulum, processed in the Golgi complex, and transported to the plasma membrane, where it is cleaved by beta secretase and gamma secretase to generate amyloid beta. RLIP, also found in the plasma membrane, facilitate the ATP-dependent transport of glutathione conjugates, drugs, and toxins. It interacts with the clathrin adapter protein to aid in clathrin-dependent endocytosis, suggesting that RLIP and APP may be both transported via endocytic vesicles. Our in vitro and in vivo studies suggest that overexpression of RLIP reduces the toxic effects of mutant APP and mutant tau. However, the exact mechanism of how RLIP affects these proteins was unclear. To investigate this, we performed co-immunoprecipitation and co-localization studies with wild-type APP, wild-type tau, and mutant tau. Our results showed that mutant APP and mutant tau interacts with RLIP while wild-type does not. In the co-localization assay, we found that RLIP co-localizes with mutant tau and mutant APP, confirming the co-immunoprecipitation findings. Importantly, the interaction between RLIP and mutant APP mutant tau leads to decreased levels of RLIP and disrupts different cellular and molecular functions in AD. These findings suggest that there is a connection between RLIP and AD. Like I said before, in AD and other neurodegenerative disorders, oxidative stress and free radicals play a major role in damaging brain cells. Reactive oxygen species like 4-hydroxynonitol are produced when mitochondria aren't functioning properly, creating a harmful cycle that causes more cell and DNA damage. This is especially troubling in AD, where existing treatments can't break down the plaques and tangles that lead to this free radical damage. One potential solution is to activate enzymes that can break down harmful lipids like 4-HNE. These enzymes are controlled by NRF2, a protein that helps increase the brain's antioxidant defenses and is essential for detoxifying harmful substances in the brain. Under normal conditions, KEAP1 binds to NRF2 in the cytoplasm, marking it for degradation by the proteasome. However, when oxidative stress occurs, KEAP1 is inactivated, allowing NRF2 to accumulate and become phosphorylated. Phosphorylated NRF2 then translocates to the nucleus, where it binds to antioxidant response elements in the DNA. This binding triggers the expression of genes that promote detoxification and antioxidant production, helping protect cells from the damaging effects of oxidative stress. This pathway is crucial for maintaining cellular health and countering the damage caused by reactive oxygen species, which are especially abundant in neurodegenerative diseases like Alzheimer's. The NRF2 pathway is tightly regulated by proteins like KEAP1 and GSK3 beta, which controls NRF2's activity. In AD, studies shown that NRF2 fails to properly activate in the brain, leading to increased oxidative stress and neurodegeneration. According to some studies, RLIP knockout mice exhibit several key physiological disruptions, including excessive oxidative stress, altered mitochondrial structure, and impaired clathrin-dependent endocytosis. Interestingly, despite an increase in NRF2 expression in response to heightened oxidative stress, these mice show lower activity in important detoxifying enzymes such as aldose reductase and glutathione peroxidase, as well as reduced glutathione levels compared to wild-type mice. This paradox may arise from a deficiency in RLIP. By impairing the body's ability to manage oxidative stress and toxic lipid metabolites, this RLIP depletion and its downstream effects on detoxification pathways may contribute to the development of neurodegenerative conditions, including AD. Now coming to how RLIP protein abnormalities are associated with mitochondrial dysfunction. Neurons, which consume up to two-thirds of their energy through sodium-potassium ATPase, heavily rely on glucose to maintain their high metabolic demands. Abnormal mitochondrial function, particularly dysfunctional mitochondrial dynamics and impaired energy production, plays a significant role in neurodegeneration. Mitochondrial dynamics, which involve the balance between fission and fusion, is crucial for proper mitochondrial function, but in AD, this balance is disrupted. Our lab findings have shown that in AD models, including postmortem human brains, there is altered expression of mitochondrial proteins like DRP1, MFN2, OPA1, and FISH1, leading to fragmented and dysfunctional mitochondria. The RLIP protein interacts with DRP1 to regulate mitochondrial division, and its dysfunction can worsen mitochondrial fragmentation, reduce ATP production, and increase reactive oxygen species leakage, all contributing to neuronal death. Moreover, RLIP is involved in mitophagy, the process that clears damaged mitochondria. In AD, decreased mitophagy due to reduced RLIP levels leads to the accumulation of defective mitochondria, which compromises cellular energy metabolism and contributes to neurodegeneration. Notably, mitophagic proteins like PINK1 and Parkin are downregulated in AD models, further impairing mitophagy. From our lab findings, RLIP also interacts with mutant tau and APP, disrupting its function and reducing its levels in AD, leading to mitochondrial dysfunction. Our previous in vitro studies shown that overexpressing RLIP significantly improved mitochondrial function, oxidative stress responses, mitophagy, mitochondrial dispersion, and biogenesis. This overexpression also enhanced mitophagy by restoring the levels of key proteins involved in this process, like PINK1 and Parkin, while reducing mitochondrial fragmentation, indicating better mitochondrial dynamics. Additionally, RLIP overexpression improved synaptic function by increasing the levels of both presynaptic protein synaptophysin and postsynaptic protein PSD95. On the other hand, silencing RLIP led to decreased cell viability, impaired mitochondrial respiration, and disrupted expression of synaptic proteins. We further confirmed these findings in an in vivo model using RLIP knockout mice. In these mice, partial silencing of RLIP resulted in increased oxidative stress, altered mitochondrial structure, and impaired clathrin-dependent endocytosis. These animals showed neurocognitive deficits similar to those observed in Alzheimer's disease model, as demonstrated by reduced mobility in the open field test, lowered performance in the Rotarot test, and cognitive decline in the Morris Watermaze test. Moreover, the mitochondrial dynamics in RLIP knockout mice resemble those in AD models, with increased expression of mitochondrial fission genes and decreased expression of fusion genes, pointing to disrupted mitochondrial function. Together, these findings emphasize the critical role of RLIP in maintaining mitochondrial health and synaptic function, with its deficiency contributing to neurodegeneration, mimicking the features of Alzheimer's disease. Now coming to RLIP's role in the PI3K AKT mTOR signaling pathway. RLIP has a significant role in regulating cellular processes like apoptosis and cell survival through its interaction with the PI3K AKT signaling pathway. PI3-AKT pathway supports various neuronal functions including synaptic plasticity, memory, and survival, and disruptions in this pathway contribute to neurodegeneration. Under normal conditions, elevated levels of RLIP activate this pathway, leading to the phosphorylation of key proteins like AKT and GSK3 beta. This phosphorylation inhibits GSK3 beta, which in turn reduces tau hyperphosphorylation and the formation of neurofibrillary tangles. By regulating this pathway, RLIP helps prevent neuronal cell death, maintaining synaptic plasticity, and promote overall brain health. However, when RLIP levels are reduced, as seen in neurodegenerative diseases like AD, GSK3 beta activation occurs, leading to tau hyperphosphorylation and neuronal apoptosis. This imbalance in pro and anti-apoptotic proteins triggers neuronal death and contributes to AD progression. In addition to tau regulation, RLIP has been shown to mitigate oxidative stress by reducing ROS levels, which are often elevated in AD. By stimulating the PI3K-AKT pathway, RLIP may help prevent neuronal damage and protect against AD, making it a promising target for therapeutic interventions aimed at enhancing neuronal survival and slowing disease progression. Now coming to RLIP's involvement in JAK-STAT pathway. In healthy cells, the JAK-STAT signaling pathway plays a critical role in promoting cell survival by preventing apoptosis. When JAK is activated, it phosphorylates STAT3, which then translocates to the nucleus as a dimer, binding to specific gene promoters. This activation of STAT3 leads to the expression of anti-apoptotic genes like BCL2, which helps reduce cell death and promote cell survival. However, in neurodegenerative diseases like AD, STAT3 activation is impaired, leading to increased neuronal apoptosis. Research in cancer cells has shown that constitutive activation of STAT3 upregulates anti-apoptotic proteins such as BCL2 and MCL1, preventing apoptosis. In contrast, when RLIP is depleted or its signaling is blocked, the phosphorylation of JAK and STAT3 is reduced, leading to decreased expression of these anti-apoptotic proteins and triggering apoptosis. This mechanism suggests that RLIP may have anti-cancer effect by inhibiting STAT3 signaling in melanoma cells. Interestingly, in the context of neurodegenerative diseases like Alzheimer's, RLIP may play a protective role. By enhancing RLIP levels, the phosphorylation of JAK and STAT3 can be upregulated, potentially reducing neuronal cell apoptosis and slowing the progression of diseases like AD. This hypothesis underscores RLIP potential as a therapeutic target for protecting neurons from death and preserving brain function in neurodegenerative conditions. These are the proposed crossbreeding of RLIP transgenic and RLIP knockout mice with different AD mouse models. To explore the role of RLIP in AD progression, we propose a series of crossbreeding experiments between RLIP transgenic mice and RLIP knockout mice with popular AD models like late onset APPKI and early onset 5XFAD as well as tau mice. Based on our lab findings that RLIP overexpression rescues AD-like features, we hypothesize that crossbreeding RLIP transgenic mice with these AD models will reveal beneficial effects against AD pathology. Furthermore, our lab previous research using heterozygous RLIP knockout mice which exhibit AD-like features such as mitochondrial abnormalities, elevated oxidative stress, and cognitive deficits, suggesting that partial RLIP depletion accelerates AD progression. Therefore, crossing RLIP knockout mice with APPKI or 5XFAD models will allow us to better understanding the mechanistic role of RLIP in AD progression. Specifically, we aim to explore how RLIP affects AB pathology, mitochondrial function, and synaptic degeneration in late onset AD with APPKI mice, and the contribution of RLIP in neuroinflammation, microgliosis, and early AD pathogenesis with 5XFAD mice. Similarly, crossbreeding with tau mice will clarify RLIP's connection to tau hyperphosphorylation and neurofibrillary tangle formation in AD. Through these experiments, we expect to identify the functional significance of RLIP in various AD-related pathways, offering a deeper understanding of how this protein contributes to neuronal health, disease progression, and potential therapeutic strategies. By investigating RLIP role in AD across different models, this research may ultimately highlight RLIP as a promising therapeutic target. Thank you for the listening. We greatly appreciate your attention and interest in our research. For any questions or further discussion, please reach us at reddylab@tthse.edu. Thank you.